Hello everyone, welcome to Biomics. Today in this video we will discuss about the eukaryotic translation. So let us first see what are the requirements for a eukaryotic translation. The first thing which is required is the mRNA. The mRNA which is present, it is having a 5' end and a 3' end. In the 5' end there is a protection that is known as 5' cap. And in the 3' end there is poly A tail. Why it is necessary of this 5' cap and poly A tail? Because the mRNA will be stable. It will not be degraded by some of the nucleases. The second thing which is main important in the mRNA is the Kojak sequence, which is known as the RBS, that is ribosome binding site. That is a specific site where the ribosome can come and bind. That specific sequence is known as Kojak sequence in case of eukaryotes. And the sequence is NN, G or A, NN. It is the sequence of the RBS, also known as Kojak sequence. Next to it the start site, that is AUG, which codes for methionine. Then again comes the G and N. Okay, and in case of eukaryotes, it is monocistronic. It is not polycistronic. So what do you mean by monocistronic? It means a single protein will be synthesized from a single RNA. Next, the next component is the ribosome. In case of eukaryotes, it is of ATS ribosome, and according to the molecular weight, this ATS ribosome is again divided into two parts. That is, the last subunit is 60S and the small subunit is 40S. In the 60S, it is having the ribosomal RNA as well as proteins. The 60S subunit is having 5.8S rRNA, 5S rRNA, 28S rRNA. Okay, and 49 proteins are present in it. In case of 40S subunit, it is having 18S rRNA and 33 proteins. The small subunit which is responsible for binding to the ribosome, it is this 18S rRNA is complementary to the mRNA. Next is the tRNA. This tRNA is having a acceptor site, that is in the 3' end it is having a acceptor site which is coded by ACC. Here the amino acid will come and bind, and this amino acid is specific. A specific tRNA is present for specific amino acid. It is decided by the anticodon part. This tRNA is having an anticodon loop which is complementary to the mRNA. So now let us see how the process of translation takes place in eukaryotes. There are two machineries in case of eukaryotic translation. In one part the ribosome is assembled with some of the proteins, and on the other part the mRNA is assembled with various type of proteins. So we will first discuss about the mRNA. The mRNA is having different type of proteins like EIF4E, EIF4G, EIF4A and EIF4B. There are certain function of these proteins. This EIF4E which is present, it comes and bind in the 5' cap site. And we can see that there is a loop present in it. If there is loop, it needs to be removed before the protein is being synthesized. Then only the ribosome subunit can scan this mRNA. Okay. So the next subunit, so the next protein is EIF4G. This EIF4G, it attaches with the E site and it connects with the A site. Okay. The next is the EIF4A. What is the function of EIF4A? After EIF4A comes the EIF4B. The main function of EIF4A is that it recruits the EIF4B and it activates or it enhances the work of EIF4B. The work of EIF4B is that it is a helicase, it helps in removal of the secondary structure or the loop for the initiation of the translation to take place in case of eukaryotes. Okay. Now, the next part is the ribosome. This ribosome is having three sites. What are the three sites? A site, B site and E site. A site is the amino acylation site, B site is the peptidal site and E site is the exit site. In case of eukaryotic translation and prokaryotic translation, it is similar. So the other factors which comes and bind is, the first thing is that this small subunit and last subunit will be dissociated first. Next, the EIF1. EIF, we can see here that it is EIF1 comes and bind in the E site. Along with EIF1 comes the 5. It is the additional. It is similar with initiation factor 1 of prokaryotes. The last subunit, there is a large protein which is EIF3. It It binds to, it touches all the proteins in the ribosome. The next is EIF1A. The EIF1A comes and bind in the A site. Okay, it comes and bind in the A site and does not allow the first tRNA to come and bind in the A site. The next is EIF2. This EIF2, it is a GTPase. It requires GTP, and the work of EIF2 is it brings the first tRNA and binds in the P site. This tRNA, which brings the amino acid, it is methionine. It is not formilated methionine, which was in case of prokaryotes. And now it forms a 43S pre-initiation complex. The ribosome with its proteins, it forms a complex which is known as 43S pre-initiation complex. Then this mRNA and the ribosome, it forms a complex. Okay, this is how it forms a complex of mRNA and the ribosome. Now, in case of eukaryotic translation, the initiation, the G protein, which was initiation EIFG, initiation factor G, which was present, it binds to the poly A tail as well as poly A tail as well as some of the proteins, poly A binding proteins. Here we can see that it is binding with a poly A binding protein. Here by two bonds they are binding with poly A proteins, and it is also binding with the poly A tail, and it starts the initiation and of the translation in a loop like manner. Okay, so next we will see what if there is no 5 prime cap in the 5 prime end. There is a sequence known as IRES sequence which bypasses the normal requirement of initiation of translation. So what was the requirement of initiation of translation? There was some proteins which was associated in the mRNA, and there were some other proteins which are associated with the machinery, of which forms the 43S pre-initiation complex. So it does not allow EIF4E to come and bind. Okay, so this IRES sequence, it bypasses the normal requirement of initiation complex, and it allows by using the EIF4G, it allows the 43S pre-initiation complex to directly come and bind in the AUG site or start site, and it forms a 60S subunit join by joining the ribosome and the mRNA, and forms the initiation as well as the elongation steps. Okay, so let us see how the eukaryotic translation is taking place. Here the machinery, that is the ribosomal machinery and the mRNA machinery, is now ready. Now the ribosome, it scans the AUG, that is the start codon site. When it enters near the AUG, what it releases is it releases the EIF2 in the form of GTP. In case of eukaryotes, the EIF2 which was present, which was bringing the first amino acid, it is already done in the ribosomal machinery. So it now only scans the start codon, and it deposits this mRNA, deposits this amino acid. Then it releases the initiation factor 2, eukaryotic initiation factor 2, in the form of GDP. Then the other factors like EIF1, EIF5, EIF4B is also removed. Why? Because this EIF1 was now the EIF1 was in the E site, that is the exit site, which was not allowing the last subunit to come and bind. Now as it has already reached the start codon, now the last subunit can come and bind. Along with EIF1, EIF5 was working. Okay, next the work of EIF4B was that it was removing the secondary structure or the loop. It was working as a helicase. Now everything is done. The loop has been removed. The mRNA is now in a linear X structure. So the work of this EIF4B is done and it is removed. After that, it recruits another one initiation factor, which is known as EIF5B, which is again a GTPase. So what is the work of EIF5B? This EIF5B, which is present, it help in the recruitment of 60S subunit, which is the last subunit in case of eukaryotes, and it helps in removal of EIF1A, which was in the A site, by which it will allow the other other amino acid or the other tRNA to come and bind in the A site. Here we can see that now the A site is after the removal of EIF5B with the help of GDP and EIF1A, the A site is now free. So this is now ready for the elongation phase to enter. Here we can see that now this A site is now empty. Okay, this A site is now empty, but it was in case of prokaryotes. In case of prokaryotes, or the first amino acid was formilated methionine, but in case of eukaryotes, it is only methionine. We can here see that in case of eukaryotes, the next tRNA is bought by EFTU, which is now in the GDP form. So in case of eukaryotes, the compensation for EIF2TU is EEF1. Okay, which brings the second tRNA and binds in the A site, and the peptide bond is transferred to the A site. Now, after bringing the tRNA to the A site, the EFTU is removed in the form of GTP. Okay, so here the other amino acid is present now, and from the peptide bond from the P side, this amino acid is transferred into this amino acid, and there is formation of peptide bond. Okay, so now there are three mechanisms to ensure that there is correct pairing between the tRNA and the mRNA. So if there is no complementary reason, how can they bind? This anticodon loop which is present of the tRNA, it should always bind with this mRNA in the correct pair. Here A is complementary with U, C is complementary with G, G is complementary with C. So this is the correct codon. If here there is G and here it is U, it cannot bind, it is not the correct binding. Here all the three anticodons and the codons should bind in similar way. Every three nucleotides should bind. If there is two nucleotide binding, it is not the correct pairing. So here there is a second one. Here it is showing the correct pairing. Here the EFTU is bringing the amino acid, and that is this polypeptide chain is being transferred into the A site for the release for forming the peptide bond, and that is GTP hydrolysis of EFTU, and the EFTU is released. So when there is incorrect pairing, this factor binding center not connected. This is a factor binding center. This is a factor binding center of the A site. If the tRNA is not correct, if the amino acid which is present in this tRNA, it is not complementary to this mRNA, this cannot bind because it is not connected with the factor binding center. So there will be no GTP hydrolysis, and this EFTU will be released in the GTP form. There will be no GTP hydrolysis, and there will be no transfer of the amino acid. Okay. Now the third one. If there is correct pairing, this tRNA will be attached to the A site, and if it is the incorrect base pairing, the tRNA cannot bind to the A site and the tRNA will be removed. This was all about how the correct tRNA should be paired with this A site. Okay. Now the work of EFG. In case of eukaryotes, or EFG is known as EF2, elongation factor 2. It is known as elongation factor 2. So EFTU was known as elongation factor 1 in case of eukaryotes, and EFG was known as elongation factor 2 in case of eukaryotes. This what is the main function of this EFG? Or we can tell that in case of eukaryotes it is EEF2. So the transfer of the tRNA from A site to the P site is done when there is little bit dissociation of this ribosomal subunit. So the EFG in the form of GTP, it comes and bind in the A site. Due to this, there will be a little bit of rotation of this ribosomal subunit, and during that time this tRNA will be transferred from A site to the P site. In this way, the EFG which is present, it will change its structure and bind in the A site, and after that it will hydrolyze. It was in the form of GTP, then it will be, when it changes its shape, it transfer into GDP form. ATP is hydrolyzed in this way, and from here the GDP will be removed, and from the exit side the removal of the tRNA take place. Okay, now next we will see what was the function of EFTS. This EFTS was working for converting the EFTU from the GDP to the GTP form, so that it can take the activated amino acid tRNA. Now the next is about the release factor or the termination phase. In case of prokaryotes, it was having RF1, RF2 and RF3, but in case of eukaryotes, it is having RF1 and RF3. RF1 and RF3 is present. In case of prokaryotes, what was that is, it was helping, RF3 was helping in dissociation of the polypeptide bond or release of the polypeptide bond. But in case of eukaryotes, the EIF3 is taking the ERF1 to the A site where there is stop codon. It recognizes the stop codon. Here we can see that ERF1 is coming and binding in the A site. It is having a GGQ sequence, and this GGQ sequence is helping in the dissociation of this amino acids. Okay, now the EIF3 is removed after the binding. After the binding, the EIF3 is removed in the form of GDP, and GGQ sequence is present here, and the ERF1 is also present here. This EF1, when it is present in the A site, and after the removal of ERF3, it binds, it recreates the RLI1. This RLI1, it helps in hydrolysis of the peptide bond. Its work is peptide hydrolysis, and it will hydrolyze the peptide bond, after which the mRNA will be dissociated from the ribosome. The last subunit and small subunit of the ribosomes are dissociated, and the RLI1 is also dissociated. This was all how the process of eukaryotic translation takes place, what are the components required for it, and what are the phenomena, what are the changes, what is the difference between the prokaryotic translation and the eukaryotic translation. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.